So what we're about to start talking about is how neurons work. And so we're going to have to talk about things like ions, and we're going to have to talk about things like electricity. Before we do that, though, let's talk a little bit about the membrane. So first of all, neurons. When you talk about them, neurons have some characteristics. One is that they're said to be irritable. An irritable structure is something that if you poke it, if you do something to it, it's going to respond. And so that's exactly what neurons do. They respond to stimuli. The way they respond to a stimulus is they produce electricity. And that electricity is called an action potential. Sometimes it's called a nerve impulse. And that action potential can travel for long distances, and then it allows these neurons to communicate with other cells. And then when you get to the end of the neuron, what's going to happen is we're going to release chemicals, and it's the chemicals that actually do the communicating. So if you look at a neuron, we already looked at it, there's the cell body, and here's the dendrites and all that, and then we have this long axon, and then we have these telodendria, and remember there are little end bulbs at the end of the telodendria. And so if we somehow stimulate this neuron, so something is going to provide a stimulus here, and we just saw one that happened. Remember, there was a glass of water, and that was the stimulus. And so then that what happened was that information gets turned into, first of all, this is light. We're going to somehow turn that information into electricity. And that electricity is going to travel down the surface of this neuron all the way till we get to where we're going. Well, remember, this one was going into the brain. And so here's our brain over here. And the brain is going to make decisions about this information, whatever it is. Well, the way the cell is going to talk to the brain is it's going to release these chemicals. And these chemicals have a name, and you may have heard of this name before. They're called neurotransmitters. And so we're going to spend time talking about those later on as well. And so that's how we somehow got from a glass of water, here's our glass of water over here, all the way to having a decision about what to do with the glass of water. And then we saw the same thing in the other direction. So here was our neuron over here in the brain, and then it went to a skeletal muscle. And so here's our skeletal muscle right here, and the skeletal muscle, then we've got to somehow tell it to contract. And so the exact same thing is going to happen again. We're going to create an electrical potential, and here the electricity is going to travel down this neuron. And when we get here, we're going to release a chemical, another neurotransmitter. The neurotransmitter is going to interact with this muscle, and then the muscle is what's going to contract to lift up the glass of water. And so, again, this is how it's going to work. And so all neurons work basically exactly the same way. There's going to be a stimulus. We're going to somehow react to the stimulus, and we're going to produce this electrical potential, and then we're going to release chemicals, and the chemicals are going to interact with other cells. But in order for us to do that, we have to talk about the membrane, because this is where it happens. It all happens on the membrane. And so hopefully from general biology, you remember a membrane. Remember, a membrane is a phospholipid bilayer. Looks sort of like this. And so these phospholipids, remember, they have a hydrophilic portion and a hydrophobic portion. They have these tails and all this stuff. But there are other things to the membranes too. There's sugars. So we have these sugars that stick off the membrane. They're oligosaccharides. We also have cholesterol inside the membrane, which helps to stabilize it. Hopefully you remember all that from general biology. But then there was also proteins in the membrane. And these proteins have a lot of different functions. Proteins can act like receptors. They can be enzymes. Remember, enzymes are going to catalyze reactions, make them happen faster. Proteins can be just structural. They're part of the membrane itself. They can be markers to let us know that these cells belong to us. Well, all of those things are important. But the things that are most important for us are these last two, because these are the things that are involved in producing these electrical potentials. And then we'll come back later on and we'll talk about some of these other things as well. But for now, let's look at this. Two things that are two functions that membranes can have in the plasma is they can be pumps. Pumps are just carriers. And then they can also be ion channels. And ion channels are basically just like pores, holes in the membrane. So let's look at these. Let's look at ion channels first. So look, there's our phospholipid bilayer right there. There it is. And then here's this protein. But this protein goes all the way through the membrane. Remember, that's called an integral protein because it goes all the way through. But look, it's hollow. There's a hollow space in the middle of this protein. It's like a tunnel through the membrane, a pore or a hole in the membrane. And what this does is it allows these ions to move, and the ions move through the channel. Now, ions move by diffusion. And that's something else you should remember from general biology. Remember, diffusion is when substances move, and they only move one way. They only move from high concentration to low concentration. They move down their gradient. It's kind of like water flowing down a hill. They go down their gradient. Well, if we look at here and we count how many ions there are out here, look, there's more ions on this side of the membrane than there is on this side. So in other words, we have a high concentration on this side, and we have a low concentration on this side. And so they're going to diffuse. But these things can't go through the membrane. They're too big, or they have the wrong charge to go through the membrane. And so if we want them to get through, we have to provide like a doorway. And that door is this ion channel. It's an open space through which it can travel through so that it can go down its gradient. Well, diffusion, remember, is passive. It does not require any energy, so it does not use ATP. It just uses this diffusion. Does not use ATP. It doesn't cost the cell anything at all. But what's happened is they move through here. Well, ion channels are just holes in membranes, but they're special holes. There are different types of these ion channels. One type of these ion channels are called leak channels. The reason they're called leak channels is things sort of leak through them very passively. Leak channels are always open. They're always open, which means that stuff can always go through them. But not just anything will go through them. They're very specific. And usually only one thing will go through them. So look, we have an ion channel which allows sodium ions to leak through the membrane. And so those are called sodium leak channels. But look at this, we also have potassium leak channels. And potassium leak channels won't let sodium go through, and sodium leak channels won't let potassium channel or go through. So we have a special channel on each side. On each membrane, there's a special channel, and they're different from each other. So sodium can only go through a sodium channel. And potassium can only go through a potassium channel. They're very specific. Well, you find leak channels on all parts of the cell. So you find them on the dendrites, you find them on the cell body, and you find them on the axon. In other words, all parts of this cell have these little leak channels that allow those ions to leak through the membrane. So if I were to blow this up, we would see on this membrane something that looks like this. So here again is our membrane. There's our phospholipid bilayer. Here's our cholesterol. There's our sugars. These are proteins. And this protein is hollow. And look, there is a pore. And this one only allows sodium to go through. So this is a sodium leak channel. Not only does only sodium go through it, but remember it only goes from high to low. So even without seeing ions here, we know that there's more over here because it's leaking this way. And it'll continue to leak until we have exactly the same amount of sodium on both sides. And after that, there will be no net movement. Does this make sense to everyone? Does everybody understand what leak channels are? So again, they're always open, which means they don't have a door on them. So when you look at a leak channel, there's no door. It's always open. So anytime the sodium is higher on one side or the other, it can leak through. And it can leak in both directions. It's just always going to go from high to low. So again, here's a sodium leak channel. And because of the way the sodium is going, I know that the concentration of sodium is higher out here, and the concentration of sodium is lower in here. Does it make sense? Okay, but not all channels are leak channels. Not all channels are open all the time. Some channels have gates on them, or doors. And if you have a door or a gate, it could be open or it could be closed. So if you look at this one, look, there's a door right here. And that door is closed. Well, if it's closed, nothing can get through it. But when the door opens, ions can move. And remember, ions only move from high concentration to low concentration. And then don't forget, just like these other channels, they're very specific. So we have gated sodium channels, and we have gated potassium channels. There are others too. There's calcium channels. There's all kinds of them. But for us, these are the ones we're mostly going to be concerned with. So I don't know which ion this is, but let's pretend it's sodium. So if this is sodium, then only sodium can go through this channel, not potassium. And when it's closed, sodium cannot go through. It can't cross the membrane because it can't make it through here. It's not until the channel opens that the ions can move. And something is going to cause this channel to open. So if we look at this picture, we can see both types of channels. So look, we have sodium channels and we have potassium channels. So the pink balls are sodium, and these purple balls are potassium. So that means this is a sodium channel. It's gated, and look, the gates are closed. So is this. But this is a potassium channel. And again, the gate is closed. But if we allow these gates to open, if this gate opens like this, what's going to happen is these sodium ions are going to go through it. And because there's more sodium out here than there is in here, the sodium is going to go in this direction. Does that make sense? But look, these are potassium channels. And so sodium is not going to go through those. Only potassium is. So if we open this channel, potassium's going to move. But which way is potassium going to go? Is it going to go from top to bottom or bottom to top? Which way is it going to go? Well, there's more potassium down here than there is in here. And so if I open the channel, potassium's going to go this way. Potassium's going to go this way. Well, what do you think would happen if I opened both channels at the same time? So I'm going to open these sodium channels and I'm going to open these potassium channels. They're all going to be open. So sodium's going to move. Well, sodium's going to move this way. It's going to go through this channel and it's going to go this way. Through this channel and it's going to go this way. But the potassium's also going to move. But the potassium's going to move this way. Because the only way the ions go is down their gradient. And the gradient of one ion has nothing to do with the gradient of the other ion. So the gradient for potassium is from outside to inside. Look, it's from extracellular into the cytoplasm, outside to inside. But the gradient for potassium is inside to outside. Does it make sense? Any questions? So there's a potassium channel and there's a sodium channel. Well, remember these gated channels have doors on them. They have gates on them. And a lot of times doors have locks. And that's exactly what's true here. These gates have locks on them. And when we think about locks, we also think about keys or some way to open the door. And so they also have these keys. And so we have three types of channels that we're going to talk about today. And one is gated channels that open with a chemical. So in other words, a chemical opens the channel. They're chemically gated channels. Or we can have mechanically gated channels. Mechanical means movement, pressure, pushing, something like that. Movement opens the channel. And then we also have voltage gated channels. And voltage is electricity. So electricity is going to open the channel. So when we look at these, what we're going to see are all three types. So if we look at chemically gated channels, look, there's a chemical. The chemical is called a neurotransmitter. We already mentioned that already, a neurotransmitter. And the neurotransmitter is going to bind right here, and it's going to make this thing open. And you can see that happen right here. Look, it bound. And when the chemical binds, the channel opens. And when the channel opens, ions can go through it. And then if you look at mechanical gated channels, remember that has to do with pressure, movement, pushing or something. Well, look, there's no pressure here. And when there's no pressure, it's closed. And the ions cannot go through it. But if I apply pressure, it changes the membrane, the ion opens, and ions can flow. And then finally, we have voltage-gated channels. So look, this is a voltage. It's like a voltmeter. Look, there's two electrodes, one on the outside, one on the inside. We're going to measure a voltage. Voltage is measured in volts. But we can also have millivolts. Milli means thousandth. So it's not 1 volt, it's a thousandth of a volt. So it's not very big, but look, this one has 70 millivolts. So that is 0.07 volts. But if I change that, look what happens. I go from minus 70 millivolts. I changed it to 60. And look what happens. At 70, the gate is closed. Nothing can go through it. But when I change it to 60, look, the gate opens and ions flow. But if you look at this channel, it has two gates. There's a gate and there's a gate. And they operate independently at different voltages. So at 60, this one opens. But look what happens if I change this to 30. This one closes. Now, if you notice, there is a positive or a negative on here. Because what we're measuring is the difference between here and here. And we'll get more back to that in a little bit later on. But look, at minus 70, they're both, or this one's closed, this one's open. At minus 60, they're both open. But if I go to plus 30, this one is closed and this one's open. So if you look at chemical gated channels, it's a chemical. It's a neurotransmitter. And the neurotransmitter is going to bind to the channel, and then the channel is going to open. So look, there's the neurotransmitter. Right there's a little receptor where it has to bind. It's like a lock and a key. So you put the key in the lock, and the channel opens. And then ions can move, just like this up here. Put the key in the lock, and the channel opens, and ions can move. If we look at mechanical gated channels, remember, pressure, movement, pushing, something like that. And what's going to happen is, when you have no pressure, look, it's closed. But when you apply pressure, the channel opens and ions flow. Same thing up here. And then finally, voltage-gated channels. Remember voltage? Voltage is a measurement of electricity. Just like when you have a battery. If you think about a battery, you could have a 6-volt battery, a 9-volt battery. And what a battery is a separation of charges. And the voltage tells you how much separation there is. So when we look at voltage-gated channels, there's a different voltage, different charge on the two sides. And look at this voltage, which is minus 70. This thing's closed. Look, it's plugged up. It's closed. But if we change it to this, look, the channel opens. And when the channel opens, ions can flow. Any questions about channels? The other really important protein on membranes are pumps. Well, think about a pump. We use pumps for all kinds of things. Look, you could have a water pump. What do you think a water pump pumps? What does a water pump pump? It pumps water. And look, if I live at the top of a hill, here's my house up here, and down here is a lake. Well, by itself, this lake water is never, ever, ever going to go up this hill. It just won't go that way. Water goes downhill. But look, if I put a pipe, I could pipe water up to my house. But a pipe's not enough. I have to put a pump down here, a water pump. And the water pump is going to use energy. So I'm going to have to plug it into something, or I'm going to have to use like gas or something like that. I'm going to have to use some sort of energy to get that water to go up that pipe and go to my house. And if I turn the electricity off, it's not going to work anymore. Cells have pumps too. And these pumps pump different things. One of the main things they pump is ions. They're active transports, not passive, it's active. There's sodium pump and water pump. Well, they're two different things. Water pumps are something we might use in our house. We don't have water pumps on our cells. But anyway, let's continue. So, pumps are active transport, which means that they use an ATP. And just like I can pump this water up a hill to my house, in other words, I can pump against the gradient, because remember, water just wants to roll down a hill, but I can pump it this way against the gradient. I can do the same thing in cells. But what I'm pumping is not water, it's ions. And there's two ions, just same two we've been talking about. And there's a pump that pumps those ions. So this pump is called the sodium-potassium ATPase pump, because what it pumps is sodium and potassium, and it uses energy in the form of ATP. But this pump does not pump in 1-to-1 ratio, and it doesn't pump in the same direction. So if you look at this pump, it looks like this. There's a place for ATP to bind. So ATP is going to bind here, and we're going to split the ATP and use the energy to power this pump. So Zoe says, can you say what is the difference in function between pumps and channels? Sure. Pumps are active transport. Remember, channels, whether they're gated or whether they're leak channels, are passive. Well, look, there's no ATP involved here. And since there's no ATP, they can only move from high to low. They can only move down their gradient. But if I had a pump, let's pretend this is a pump. It's not, but let's pretend it is. A pump pushes things. So it pushes, and so it can move against a gradient. And because it's pushing, it requires energy. And that energy is in the form of ATP. So look, this is a sodium channel, and this is a sodium pump. So sodium channels only go down their gradient. It's passive. It's diffusion. Does not require ATP. It can only go down its gradient. But a pump moves against the gradient. It's active. It's active transport, and so it requires ATP. So if we go back to this pump we're talking about, the pump that we're going to be most concerned with is this sodium-potassium ATPase pump. And it looks like this. So there's this pump right here. So look, it has an ATP binding site. That's so that an ATP can come here and fit in that space. We can split it and we can get energy out of it. And we can use that energy to push sodium and potassium. But it doesn't push them in the same direction. The sodium-potassium pump pumps sodium out of the cell, and it pumps potassium into the cell. And it also does not pump in a 1-to-1 ratio. It pumps in a 3-to-2 ratio. So it pumps 3 sodiums out, and then it turns right around and brings 2 potassiums in. So it's here on the membrane. It looks like this, and it's going to pump 3 sodiums out, and then it's going to turn right around and bring 2 potassiums in. And it's going to use an ATP in order to power that or make that happen. So when you look at it, there's the 3 sodiums. It's going to pump them out. And then it's going to turn around and bring 2 potassiums back in. 3 sodiums out, 2 potassiums in. That's all it does. Cannot do anything else. And remember, every time it does that, it uses an ATP. But it can't do 2 and 1 or 1 and 5 or whatever. It's just 3 sodiums out and 2 potassiums in. It's a 3-to-2 ratio. So if we look at it, see if I can find another picture. Well, there's that pump. I guess I don't have one. But again, this is all it can do is pump 3 sodiums out and bring 2 potassiums back in. But look, not only is it pumping sodium and potassium are both ions. They have a charge on them. So look, when I pump 3 sodiums out, I'm pumping 3 positive charges out. And when I bring potassium in, It also has a positive charge, but I'm only bringing 2 positives back in. So look, 3 positives out, 2 positives in. 3 positives out, 2 positives in. And it happens over and over and over again. 3 positives out, 2 positives in. 3 positives out, 2 positives in. Well, look what's happening here. As I do that, look at this side over here, the outside. Look at it. It's more positive. And if I look in here, the inside, it's less positive. Another way to say less positive is it's more negative. And if I keep doing that and keep doing that, I'm going to wind up with a situation where it's more positive on the outside and more negative on the inside. Every time this thing pumps, it's going to pump sodium out, 3 sodiums out, and bring 2 potassiums back in. But it's also pumping 3 positives out, because each sodium is a positive, and only bringing 2 positives in, because potassium is positive too. And so if I keep doing it, sodium's going to build up on the outside, potassium's going to build up on the inside. And the outside's going to build up positives. So it's going to be more positive. The inside is going to be less positive. Or again, another way to say that is more negative. And so if I keep doing it, look what happened. I pump sodium out, sodium out, sodium out. I'm going to bring potassium in, potassium in, potassium in. And look what I get. I wind up with more sodium on the outside. Look, it's 150 on the outside. But on the inside, look, it's only 15. And I'm going to wind up with more potassium on the inside. Look, it's 150 on the inside. But it's only 5 on the outside. And then look at this. Not only are there differences in sodium and potassium concentration, look, the outside is positive. All the way around, it's positive. The outside is more positive, and the inside is more negative. Because remember, every time this pump pumps, I pump 3 positives out, and I only bring back in 2 positives. 3 positives out, 2 positives in. 3 positives out, 2 positives in. And so look, the outside becomes more positive, and the inside becomes less positive or more negative. So when I look at this situation, look what I've got. I've got gradients. I've got gradients because there's more sodium on the outside. So sodium wants to come back in. So the sodium gradient is from the outside to the inside. Potassium, on the other hand, there's more on the inside, so it wants to go out. So its gradient is from the inside to the outside. So if I open up channels, sodium is going to want to come in because it wants to go down its gradient. And if I open up channels, potassium's going to want to go out because it wants to go down its gradient. Does it make sense? So this is what it looks like. So there's more sodium on the outside, a lot less sodium on the inside, more potassium on the inside, and a lot less potassium on the outside. The outside is more positive, and the inside is more negative. Any questions? But let's don't forget about our leak channels. Remember, leak channels are always open, and they allow this passive movement of ions across the membrane. So we've got sodium leak channels and we've got potassium leak channels. So look at this. We've got this situation. Here's this sodium-potassium pump, and it's pumping 3 sodiums out and bringing 2 potassiums in. So what we're going to wind up with is more sodium on the outside. So look at all this sodium that's out here. Lots of sodium. And it's bringing potassium on the inside. So look, we got all this potassium on here. Lots of potassium. And so if I let it, the sodium will go down its gradient. Well, we've got leak channels. So these are leak channels. So look what's going to happen. As soon as I build up this gradient, I'm going to let sodium, there's a sodium leak channel right there. Sodium's going to leak back in. But I also have these potassium leak channels, and they're right here. And so potassium is going to leak back out. But as soon as that happens, this is going to reverse the whole thing. It's going to pump the sodium back in, back out, and pump the potassium back in. So it's a little bit like a fountain. Think about a fountain. You can go right now to Target or Walmart or someplace and you could buy a little fountain for your desk. So here's what it's got. It's got a reservoir, but it also has a way for the water to go up, and then the water is going to drip back in. And then when you look in here, there's going to be a little pump in here. And you got to put the pump, you got to hook it up to electricity, and otherwise it won't work. And then you pour water in the little reservoir. So you put water in here. And what's going to happen is when this starts, the water is going to go this way. It's going to get pumped out. But look what happens when it gets over here. It's going to leak back in. But as soon as it leaks back in, it's going to get pumped out. But as soon as it's pumped out, it's going to leak back in. And even though there's water circulating like crazy in here, if I look at the level of water and I watch it, the level of water isn't going to change. It's going to stay at this level right here. Because every time the water pumps out, it leaks back in. And every time it leaks back in, it gets pumped back out. And so if I measure the level of water, it goes up and down, but only just a tiny bit. because every time it leaks out, it gets pumped back in. Every time it pumps out, it leaks back in. So the water level is not really going to change. So it's not this pump right here. It's not the pump that we have that's black or whatever here, like this. This is a water pump. Well, we don't have a water pump. We have a sodium and a potassium pump. We're not pumping water. We're pumping sodium and potassium. But look, every time the sodium gets pumped out, it leaks back in. And every time the sodium gets pumped in, it leaks back out. But again, just like with our little fountain, if we look at the sodium and potassium levels, they're not really going to change because every time they leak, they're pumped. Every time they pump, they leak. So when you look at it, this is what's going on here. Here is our pump right here. And remember what it does. It pumps 3 sodiums out. 1, 2, 3 sodiums out. And then it turns around and brings 1, 2 potassiums in. But as soon as that happens, look what's going to happen. The sodium is going to tend to leak back in. So the sodium is going to come back in through this leak channel. Come back in through this leak channel. The potassium is going to tend to leak out. So every time I pump it in, it's going to leak out through its leak channel. But as soon as that happens, this is going to grab the sodium, pump the sodium back out, grab the potassium, pump the potassium back in. It's just like our little fountain, like we just saw. It has this pump, but it also allows the ions to leak. So if we look at the level of sodium and we look at the level of potassium on the 2 sides of the cell, they don't really change. And so if we look at what those levels look like, they look like, well, I guess I could go backwards. They look like this. So look, this number really doesn't change. This number doesn't change. Same thing here and here. Because again, every time it leaks out, it gets pumped back in. Every time it leaks in, it gets pumped back out. And so if we were to look at it, the levels don't really change. And so again, it's like our little fountain. There's our pump, and here's our leaking. There's our pump, and here's our leaking. So as the sodium leaks in, it gets pumped back out. As the potassium leaks out, it gets pumped back in. And so what we wind up with is sort of a steady state. Just like with our fountain, we get sort of a steady state. The water level in the fountain doesn't really change. If we measure it and look at it, it's not going to change because we got this pump going in one direction and we've got the leaking going in the other direction. And so we get a steady state between the pump and between the leaking. A steady state. Make sense? Between the pump and the leaking. And here we can see it sort of in a different view. But there's the pump. And look, it's pumping sodium out and it's bringing potassium in. But as soon as we pump the sodium out, the sodium leaks back in. As soon as we pump the potassium out in, it leaks back out. So again, we get this steady state. And what we wind up with is a membrane where there's more sodium on the outside and more potassium on the inside. The outside is positive and the inside is negative. And so if I look at these two sides of the membrane, they're very different from each other. And another way to say that is to say that the membrane is polarized. So polarized, this side, the outside, does not look like the inside. They're different from each other. They're polarized. Not only are there differences in sodium and potassium, there's a difference in charge. Remember, we can measure a charge. And so if we look at the charge across this membrane, the outside's positive, the inside's negative. The charge on a neuron tends to be about minus 70 millivolts. It's a charge difference across the membrane. And the only reason it's negative is because the first guy who did it decided to measure it inside relative to outside. So if we look at it, the inside relative to the inside is negative. So the number is negative. And then this number tells us how big a difference there is between the two sides. So there's a 70 millivolt difference between the two sides, and the inside is negative compared to the outside. So minus 70 millivolts. That's an average, but it's minus 70 millivolts. And it's a steady state. Remember, it's not going to change very much. If I graph it, it's going to stay right around minus 70, and it's just going to stay that way. It's a steady state. And that steady state we call resting potential. So resting potential is when the membrane is doing its thing. It's not doing nothing. It's doing this. We're pumping 3 sodiums out. We're bringing 2 potassiums back in. The sodium tends to leak back in. The potassium tends to leak back out. And we're going to wind up with a steady state. And if we measure the voltage across the membrane, the voltage is going to be minus 70 millivolts. And that is resting potential. So when we look at resting potential, it doesn't matter where I measure it. I can measure it here, but I can measure it here. I can measure it here. I can measure it here. We have leak channels everywhere, and we have the pump everywhere. So it's minus 70 all along the membrane. Again, this is resting potential. It sounds like nothing's going on, but remember that's not true. All of this is going on. We're pumping 3 sodiums out. We're bringing 2 potassiums back in. The sodium is leaking in, and the potassium is leaking out. But just like our little fountain, we get this steady state where the water level in the fountain, the voltage here, doesn't really change. I don't know what this level in the fountain is, but if this were the voltage, the level would be minus 70 millivolts. Does it make sense to everyone? So look, if we were to graph that, it doesn't change. It's about minus 70 millivolts. It doesn't change. So here's 0 right here. So there's minus 70. And look, it's a straight line. It does not change. That's resting potential. It's the steady state. So what produces resting potential? It's 2 things. It's the pump. Remember, it's pumping 3 sodiums out and pumping 2 potassiums in. And then sodium leaks in and potassium leaks out. But as soon as that happens, the pump is going to reverse it, and we're going to get this steady state. That's resting potential. So if you put all that together, this is what it looks like. So there's our pump, and look, it uses an ATP. Pumps 3 sodiums out, brings 2 potassiums back in. And then those 3 sodiums tend to leak back in through the sodium leak channel. And the 2 potassiums tend to leak back out through the potassium leak channel. And we get this steady state. And if we measure the voltage at this steady state, remember the outside's more positive, the inside's more negative. And if we want to know how much more positive, it's minus 70 millivolts. So this is a nice summary slide of everything that's going on. Any questions? So that's where we're going to stop for today. So before we meet again tomorrow, make sure you go over this and you understand resting potential. And understand that resting potential is the combination of these 2 things, the pump and the leak channels. Pump, again, pumping 3 sodiums out, 2 potassiums in, and then the sodium leaks back in, and the potassium leaks back out. But as soon as that gets done, this is going to pump it in the other direction, and we're going to wind up with this steady state. And if we measure the voltage across the membrane at this steady state, at resting potential, it's minus 70 millivolts. Any questions? Okay, well, that's where we're going to stop for today. So next time we're going to pick up and we're going to talk about how we can use these potentials to create signals and send the signals down the membrane so that we can talk to other cells.